My name is Gunnar Joachim Siljadal. I'm one of the conference crew here. And for this talk, we will have Professor Dunfield with a talk about the biodegradation of starch by microbial communities in soil collected from Oltupai Gorge in Tanzania. The floor is yours, Professor.
All right, thanks, Joachim. It's a pleasure for me to be here at this conference. It's been very interesting. I must say it's a long ways away from anything that I do, very peripheral to this. But I think everybody in science appreciates the importance of the Oltupai Gorge and really the history of science. So it's really an honor for me and a pleasure to be involved in this, even in a very peripheral sense. And what I'm going to talk to you today is a work from an MSc student who recently graduated from my laboratory, Tope Akaju. He's the first author here. And this was a collaborative project with Julio Mercader, who most of you have already seen during the course of this conference. So Julio is obviously big on multidisciplinary research, so that's how we became involved in this. The other people involved here are Angela Smirnova, who was my lab manager, who taught Tope pretty much everything he did, and of course, Julio. So why are we doing this? Why are we studying biodegradation of starch by microbes in soil from the Oltupai Gorge? This grew out of work that Julio had been doing, looking at preserved starch granules on stone tools. These granules can be preserved in the archaeological record, and they can be used as an indicator of ancient diet, tool use, and agricultural activities. Pardon me. But an ongoing question is how a labile and easily degradable molecule like starch can survive in the archaeological record for thousands or millions of years. That's still a bit of a mystery. And what happened is that Julio came to me one day. He knew I was a soil microbial ecologist, and here we are down by the schoolyard, and he was asking me, can we learn anything about the preservation of starch granules in soils on stone tools or other articles for thousands or millions of years from a study of microbial starch degradation? And I thought, and I said, well, probably not, but who knows, maybe we should just do the experiment and see what we find. It's the kind of thing I like to do anyway, as a soil microbiologist. So we'll get something out of it, and maybe it'll help you out. Of course, what I was thinking on the side there was, where am I going to find a student who will follow an experiment for 1,000,000 years or so? Well, here's the student. This is Tope, working in the laboratory. Sadly, he was a great disappointment to me because he only managed 2.5 years rather than 1,000,000. But actually, he did a fantastic job and finished up his thesis, and is now a productive member of society, so he can't give this talk, which is why I'm giving it for him. And this is what he found in those 2.5 years. His research objectives were to understand microbial communities that degrade different starches over time using molecular DNA-based methods. And I'm going to give you a primer into DNA-based microbial ecology, everything you need to know in just a couple of minutes. To determine the most suitable conditions for the enzymatic degradation or preservation of starch granules in a medium like soil, and to examine the morphological changes in starch grain structures under different biodegradation conditions and different times. So here's my 2-minute primer in microbial ecology for those of you who have never done it. This is really all you need to know. The first thing you need to know is that microbes are extremely diverse. We estimate that there's somewhere between 10 million and 100 billion species of microbes that are extant at the present time. And I guess for people who work with hominids, that's a pretty intractable number. The problem is nearly all of these have never been grown in a laboratory. We've only, in the history of microbiology, only ever grown about 10,000 species. So nearly everything out there is what we call uncultured. So we used to think we knew everything. Now we know that we know nothing. And what you would used to do if you wanted to study a process like starch degradation by microbes, you would make a medium with a lot of starch in it. You would grow some bacteria on that media. you would pick out that bacteria and isolate them and then use them as a kind of a model organism to study starch degradation. And this has been a very successful approach through the history of microbiology, but it's not very realistic for two reasons. These models just don't turn out to be very good models. And the first reason is that in nature, processes are very rarely carried out by a single microbe. They're carried out by complex communities. The second reason is that the model organisms are very rarely the same organisms that actually operate in nature, because we don't know 99.9% of the things that operate in nature. So nowadays, what we do instead of this culture-based approach is to use DNA-based approaches to understand what microbes are present and what they're doing in a particular environment. So if I take a sample like a teaspoon of soil here in the upper right, that may contain thousands of different species of microbes, all doing different things, all with different ecological niches, et cetera, represented by this sort of diagram of different shapes. And what I'm going to do is extract all of the DNA from that soil and then look for what I call fingerprint genes. These are genes that every microbe has, and the sequence of that gene is a fingerprint for every microbial species. So if I take that mixture of DNA and throw it on a next generation sequencer, I can get thousands and thousands of sequencings, each one which is a fingerprint for a particular microbial species, and in that way characterize the microbes that are present and active in a particular sample. So what we did, therefore, what Tope did, the first thing we did is we had some of Julio's people collect some soil for us from the Olduvai. Sorry, my girls are homeschooling, making lots of noise, so I try to get them to stop here. So what Tope did is he took some of that soil and extracted the DNA from it. It's a very high pH alkaline soil, low moisture content, and a very low humic content in the sample we had. And what you do is you do this next generation sequencing of the fingerprint genes, and you come up with a set of usually thousands of species that you find in that sample. And in this little pie chart, I've just divided them into microbial kingdoms. So we divide all microbes into about 100 different kingdoms, each of which contains thousands of species. The most important kingdoms in this sample were actinobacteria, firmicutes, and a few others. This is actually a pretty typical community for an arid alkaline soil. If I took a soil with similar properties from North America, I would find a similar distribution of species as well. One of the giveaways is this called Gemmatomodidettis. That's kind of a signature for arid alkaline soil. So now we know what's there. The next thing Tope did was to look to see whether the microbes that occur, next to a starchy tuber, and in the starchy tuber we picked is this Epomoea longituba, kind of a sweet potato of ethnobotanical interest in the area. We wanted to see if certain microbes wanted to associate with that starchy tuber as opposed to the soil. Rather than show you the species, what we've done here is a data reduction analysis that takes the thousands of species and reduces it to a kind of a two-dimensional space here, where each point is a sample. And the closer points are together, the more similar the overall communities are, and the further apart they are, the less similar the overall communities are. And the colors here represent bulk soil, which was taken away from the tuber, and tubersphere soil, which is soil that was brushed off of the tuber itself. And I think you can pretty clearly tell by the sort of the clustering of the points based on color that there's a very particular microbial community associated with these starchy tubers. And this was true in different sites. So what kind of bacteria associate with these starchy tubers? The typical bacteria we found on the surface of the tuber were mostly actinobacteria. These are filamentous bacteria. They form a kind of a network of filaments, much like a fungi, except much, much smaller. This is a very effective way of penetrating different substrates and digesting them. Many actinobacteria are known to be starch degrading microbes. In fact, the best way to grow actinobacteria is to make something called potato agar, where you just grind up a bunch of potatoes and let the bacteria grow on it. So obviously they like starch. Two of the genera we found, Streptomyces and Nocardioides, are actually known scab-forming bacteria of tubers. And you can see a picture here on the right of scabs forming on potatoes caused by Streptomyces. So it makes perfect sense. These are associating with the starchy tubers. There were a lot of other things that we found associated with the tubers. These were often known endophytes, things that grow in between plant cells. They can be pathogens or symbiotes. And alpha proteobacteria, which is a different kingdom of bacteria, which also tend to associate with plants. So we definitely have a very distinct tubersphere community that likes to associate with these starchy tubers. The last point I've put here is that we did not find a bacterium called Bacillus associated with the tubers at all. And the reason I point this out is this is going to become important in the next experiment, as well as the fact that Bacillus is really the model organism for starch degradation in a lot of studies. And I don't think it's actually very important. So this is what Tope did next. He wanted to take this knowledge that we have of the microbial communities and see if we see something similar when we actually add ground up starch to a soil, what kind of microbes degrade that and what it looks like. So his molecular methods here are in red on the right. So he'd add some starch to soil under various conditions. I'm only going to show you one condition, essentially aerobic incubations with a little bit of water to stimulate activity. And over time, as the starch degraded, he would extract the DNA. He would look at these fingerprinting genes. And actually, we have a different fingerprinting gene for bacteria versus eukaryotes. So eukaryotes are more complex organisms with nuclei. They include things like protozoans, ciliates, plants, animals, humans. Anything more complex than a microbe is a eukaryote, but we can characterize both of them. And then with the next gen sequencing, we could characterize the communities that are doing this particular process. I've got in blue here on the right something that we're actually in the process of doing, and is probably the most relevant part of the experiment, especially to Julio. And that's at various times, these samples, we preserve them so that we can look at them afterwards and look at the starch structures that are remaining in all of these incubations and try to correlate these with important structures, for example, that Julio sees on stone implements. Okay, so for this experiment, Tope took two substrates, one which is purified potato starch, and this is essentially 100% pure starch. So all the other components of the plant have been removed, the proteins, the DNA, etc. For the other one, we took a ground up local tuber. So that's the Epomoea again, the thing we were studying for its tubersphere community. This is a little bit different because tubers are usually only 60 to 80% starch by weight. So this is going to have a few other things in it as well. It's going to have some proteins, going to have some soluble sugars, etc., but a lot of starch as well. So he ground these up and he added them to soil, and he's just going to look at what microbial communities break these starches down. And he would monitor the activity simply by monitoring CO2 production in these incubations. And the idea is, as the microbes break down the starch, they make biomass from some of it and they make CO2 from the rest of it. So by looking at CO2 production, we can see the progress of the degradation. And in fact, the amount of CO2 produced here is on the y-axis. Actually, we only added about 2.5 to 3 millimolar. So I actually got more carbon back from the Epomea than we put in. And this is actually quite normal. This is called a soil priming effect. If you get the microbes working, they'll actually start to degrade other things that are already there. The potato we didn't get all back, which is interesting in itself, and it suggests that a lot of the potato starch was recalcitrant. But the whole process sort of completed in about 100 days and then really slowed down to almost nothing. So what did he see microbially? The first thing you see in the incubation with Ipomoea, ground up Ipomoea tuber, was a rapid, incredible growth of a bacterium called Bacillus, which I've mentioned before. The y-axis here is relative abundance, and that means when we do these sequencing sets, we express the abundance of each bacteria as a relative of the total. So you see this orange line, which is leveling off at about 0.5 here. That means 50% of all the bacteria we detect in the sample is Bacillus. So it's grown from about 1% of the entire community to about 50% in approximately 6 days. So it really went crazy when we added that Ipomoea. Interestingly, it did nothing to the potato starch, however. So my suspicion is that this organism isn't really degrading the starch. It's probably growing on a lot of those other compounds, like the proteins or the sugars that are in that powder we produced. And this makes sense. Bacillus is sort of the classic microbial weed. If you give it nice, easily degradable substrates, it goes crazy. It grows like a house on fire, and then it produces spores and sits around literally for hundreds of years doing nothing. So even though this bacterium stays at a very high level, it's not necessarily doing anything. It's probably in a dormant state. So that's the most obvious thing you see from adding this. Here's, how am I doing for time? Here's actually the entire community. And again, on the y-axis, we have relative abundance, and these are stacked. So the blue is our Bacillus again. You see from time on the x-axis, from 0 to 6 days, that Bacillus increased to about 40% very rapidly in this experiment. They're stacked. So for example, the blue, which is Bacillus, is 40, and then we have a orange bar, which is Nocardioides, and that's about 10%, et cetera, et cetera. These stacks don't add up to 100 because we're not showing you everything. We're only showing you the important things. But what you can see from the Ipomoea is, yeah, we had that initial burst of Bacillus. We also had this initial burst of Nocardioides. Remember, that was one of those tubersphere scab-forming bacteria. So it obviously liked having the tuber ground up and fed to it as well. But then a few other things started to appear at later times. So for example, this light blue one is a glycomyces. This showed up slightly later, 14 to 41 days. And some of these other ones, like this dark gray Propionibacteria, Promicromonospora, they started to show up at later times. So there appears to be a kind of a succession of bacteria. Nearly all of these bacteria are actinobacteria, these filamentous known starch degraders. And on the right here is just the control soil, which didn't change over time, which proved that the changes we're seeing in the starch added treatment are really due to the starch. The potato treatment was more interesting because it was spread out, it was a bit slower. The Bacillus didn't grow, so the Bacillus really didn't like the starch, but a lot of other things did. So the yellow one here is again our Streptomyces. And again, that was one of our tubersphere scab-forming bacteria. It grew gradually and peaked at about day 41. Some of these other ones, for example, this brown one, Promicromonospora, and this gray one, Isoptericola, they peaked at slightly later times. So again, we have this kind of succession of different microbes following each other over time and involved in the degradation of the starch. And again, they're all actinobacteria, really. The other thing we did, and I'm only going to show you the potato here for time reasons, was to look at the eukaryote community. Remember, these are larger organisms, although they can be single-celled, but they're very huge single cells, things like fungi. The control soil, again, over time, didn't change a lot. The one thing you see is this light orange segment declined over time. Those are nematodes. Those are worms. And essentially, they died because we didn't give them anything to eat. So over time, they just went dead. The blue one, Gregarinaceae, is actually a parasite of the nematodes. So as the nematodes died, it died as well. But otherwise, the control community didn't change much. The one where we added potato changed dramatically, but it didn't happen as fast as the bacteria. So nothing happened really from day 0 to 6, where this bacillus and nocardioides were going crazy in one of our treatments. Not much happened for the eukaryotes, but by day 41, we saw a huge bloom of Ascomycota, which are fungi, and they're also known to degrade starch. And then after that, actually, our nematodes made a comeback, and probably because they were feeding on the Ascomycete fungi. So we have a kind of an interesting ecology going on, breaking down this starch. It's a lot of things following each other. It includes both bacteria and eukaryotes, and I've tried to summarize it here. So as if we add starch to a soil-based medium, we see a little burst of these weedy organisms, bacillus, and then a whole lot of these actinobacteria. First off, the weedy types like nocardioides, and then some slower growing ones like streptomyces, and then much later in the process, they're taken over by even more slower growing actinomycetes like Poromicromonospora. We have Ascomycota fungi contributing to this process. Usually later on, it seems they don't grow as fast as the bacteria. And then we have larger organisms like arthropods and nematodes that actually start to graze on the fungi. So we have a kind of a really complex ecology here of starch degradation. And I think the bottom line is really that starch degradation, if we want to model it, we really can't do it with a single organism as has been done in the past. We have to understand that it's an entire community, and it's actually quite a complex community succeeding one another over time. So I believe that using a pure culture, especially of a weed like bacillus, to study starch degradation is not really a realistic. I'll also mention that I just showed you essentially one experiment that Tope did. He actually did a lot of different things. He studied whether the amount of water added to the soil affected it. He studied whether the amount of oxygen in the atmosphere was important. He studied different starch types, wheat, corn, potato, epomoea. And he studied calcification, whether we could precipitate some of the starch with calcium phosphate to model something that occurs with dental calculus, and whether this protected some of the starch from degradation. And without showing you all the results, the bottom line is that, in fact, all of these things affected the rate of starch degradation, and they affected the microbial communities that were involved in the starch degradation. So all of these factors were pretty important, and we could really slow down starch degradation much more than we showed in the original experiment by drier conditions, less oxygen, precipitation with calcium phosphate, et cetera. Obviously, we're still not in thousands of years, but these things definitely slow down the degradation. This little diagram actually summarizes some of Tope's results. For time restrictions, I'm not going to explain it all in detail, but basically some starches were much faster to degrade, and some conditions caused degradation to speed up as well. So finally, coming back to my question that Julio asked me down by the schoolyard so long ago, does this help us at all to understand the preservation of starch granules on stone tools? And the answer is I still don't know. I think Tope did a nice project, and we can easily publish that because it's a good little example of microbial ecology and how complex it is, even with a very simple substrate. I think the one thing that will possibly help, and this is what we're doing now in blue, is we have all these preserved samples from the experiments that Tope did, where we followed the communities. We know which microbes are operating at different times. And we can look at those microscopically and see what kind of starch structures are being produced at different times, and whether they look at all like the things that Julio notices on the stone tools. And with that, I'm going to end up. Again, this was part of this large Stone Tools Diet and Sociality project funded by SSHRC. So thanks to the Social Sciences and Humanity Research Council of Canada. I'll also thank the Natural Sciences and Engineering Research Council in Canada. So we have three main funding groups, and this is very multidisciplinary because we have both of them involved here. And thank you for listening, and especially again, thanks to Tope, who did such a nice master's thesis, which I managed to talk to you about today.
Thank you for a fantastic presentation. We even got some appreciation from the chat saying you rock, which is...
Yeah, that disturbed me for a second. That was Julio just trying to put me off my stride, I think.
But we do have time for a couple of questions. So I think I will post what we have time for. This is kind of interesting to me personally, because I have a background in biology and microbiology myself. So I wanted to ask a personal question, and I don't know if you have encountered this, but you mentioned nematodes grazing on a lot of these microbes or fungi and whatnot. And coming from Norway, I worked a lot with Globodera rostokiensis, which is this nematode that ruins a lot of the potato farming. And we've always kind of feared it because it can remain dormant for a longer periods of time within the potato and whatnot. And I'm wondering if the nematodes that you saw in this experiment with Tope had any relation to that, or if you, because we have a traditional view that these nematodes eat and destroy the potatoes, if that was anything you experienced at all.
Yeah, so that's also a problem. We didn't see when we added starch really nematodes increasing at all. It really only happened right at the end of the sort of the microbial process. And so the species of nematodes we identified appeared to be fungal feeders in this particular experiment. And that doesn't mean, though, that under different conditions, the nematodes wouldn't be a big problem for decomposing tubers as well. I suspect they're going to prefer much wetter conditions than we were giving them. They tend to go dormant under dry conditions. But that's and you know, how's that going to affect starch too, being grazed on by larger animals as well? So many different processes going on here. I think there's a lot to unravel.
Yeah, and touching upon that, we actually have a question talking about specifically that. Do larger creatures like invertebrates also play a role in starch degradation? I think you have touched upon that.
We did see some arthropods at one point, sort of a little burst of those. And again, you know, they're so complex. You know, are they feeding on the microbes, or are they feeding directly on the starch? It could be either. They almost definitely play a role. Yes. Yeah. My knowledge base tends to stop at anything that's bigger than 10 microns, though. So that's where I lose interest. But as an ecological system, this is all very relevant. Yes, all these things are happening.
Awesome. All right. I think we have time for one last question, so I will post this one here. What would future experiments look like to expand upon this research?
There are a few things we'd like to do. So I'm making a lot of inferences here based on basically just seeing things rise and fall over time. But there are more involved experiments that we can do to unravel what's happening. So for example, one of the things I'd like to have a student do is experiment with stable isotopes, where we essentially add a 13C labeled starch to our system, and by doing that, you can actually track exactly where the 13 carbon goes through the system. And it's much more exact in understanding the ecological interactions. Another thing we'd like to do is actually get down to the nitty-gritty of the actual enzymes that are in there degrading the starch, the amylases. And again, the samples we have will allow us to do that with metagenomics. And finally, the really interesting thing, of course, the microscopy, which we've planned to do, and you know, the samples are archived, and hopefully that'll tell us something about what the starch granules look like at various types of degradation processes.
All right. Unfortunately, I think we are out of time there. Thank you so much once again. That was an amazing talk, and thank you for your answers.
Yeah, thank you.